And we'll leave our trilobite up here because trilobites are great, aren't they? So let's talk Ecdysozoa. Ecdysozoa is this group of a bunch of things. It's going to include arthropods. It's also going to have things like preapulids, the penis worms, and nematodes and nematomorphs, I think, are in there as well. And so these are all going to be organisms, as well as like insects. Yeah, all the crabs. Those are included when we say arthropods. Things like onychophorans, the velvet worm, and probably things like lobopodians, which are the little things that walked along, and some of them had spikes on their back, 2 rows sticking up like that. All right, so question is, when did this sort of shed your skin if you want to grow behavior develop? Because this is not a thing that we particularly do. Yeah, I don't shed my skin anyway, all in one go. And my skin doesn't provide a whole lot of mechanical support either. Yeah, if I'm going into battle, I better put on some actual armor because I was not born with any. Whereas some of these guys are. Sometimes it's hard, sometimes it's soft, but it's always at least leathery and somewhat protective. Um, and so the idea is that being an ecdysozoan may have been the first key to being able to survive on land. This sort of an outer dry skeleton that is essentially disposable. There's a thought that that may have been one of the components that was later reused when some of these arthropod-like things did colonize the land. So as an approach to figuring out, all right, when and how did this happen, this particular group looked at some of the genes that are heavily involved in growing new skin, shedding skin, that sort of things. The exoskeleton development of arthropods. And you can find these things in all these extant groups, and you can assume that they were probably there in things like trilobites. But with trilobites, the best we can do is try to imagine or infer their genetics. So those are not included here. These are just things for which we have data. Then if you do that, and you take it's a thing called a molecular clock analysis. It's a way you can build your tree, and you can say, all right, if I make one genetic change every week, and I've got 10 genetic changes between this individual and this one, they must be about 10 weeks since they had a common ancestor. If they make one change per week, per generation, something like that. And yeah, that's a reasonable way to do it. I mean, there's not really another great way to do it. And so what they did with this study is they said, all right, let's do the molecular clock analysis. Let's see how this aligns with what we know about fossils. So they took all these genes that are involved, and each color band is a different gene. Every little line here, they're showing, it's like the error bars on when these things diverged. And so all the ones that you find in these sort of primitive insect collembolas seem to have diverged around this time. We think that this group probably diverges back here, but those particular genes all seem to diverge at this point. And so, okay. And so on each lineage, they're looking at when these particular genes diverge. And if you go back, so the group that's our out group are going to be Lophotrochozoa. Those are things like an earthworm or a brachiopod. Those are not in the Ecdysozoa. They do not shed brachiopods do not shed their shells. Oh my gosh, it's integral. A lot of these genes go back and chronologically seem to line up in kind of the mid-Ediacaran, which means, and that's way before we find fossils of things that are confirmed Ecdysozoa. And it's way before we find things that are even like particularly very closely related to modern groups. So the idea is that some of these genes were there in at least some of the Ediacaran animals, these mystery weirdo animals, and that some of these things may have been repurposed, leading to these sort of explosion of arthropod groups and ecdysozoan groups, other ecdysozoan groups. But the divergence of things like preapulid worms, or penis worms, and chinoruncs are little tiny things. is actually all the way back pretty securely. Like the error bars do not come out of the Ediacaran. So theoretically, there's some fossil back there, or a fossil that we haven't found yet from back in that time, that's actually ancestral to all the Ecdysozoa that we know now. An interesting thing to find, if you could find it. Yeah. So that'll be a challenge for future paleontologists. So, how do we recognize an ecdysozoan when it's a fossil? Well, you're going to have lots of molts. Just as you have one body and lots of clothes, same thing for these ecdysozoa, except that it's every time they grow, they're going to molt. And so one individual can leave behind many different molts. And so molts are way more common than full body fossils in the fossil record. That's just the math of the thing. These are all different trilobites over here, like our trilobite above, our little mascot again. And these are showing what is potentially different molting behaviors. So in trilobites, we know that these things, which are called the cheeks, free cheeks, and it's kind of a break right where the eye stops, because they don't want to shed their eyes, because they worked really hard on building the lenses and stuff for those eyes. So they will generally keep those from molt to molt. But they'll shed the stuff around them, they'll shed the stuff in front of them, they'll shed the stuff over them and behind them, and get rid of all that stuff. A little covering on the outside can go. But you got to keep those important parts then, like the organs inside. So we got the cheeks coming off, and then this one probably crawled right out the front because the front's missing. And the rest of it is very intact. Yeah. In this case, we've got two breaks. So we've got a trilobite that may have crawled out over its own cephalon, but we've got another break here by the pygidium. And so it broke, the exoskeleton broke in a couple places for it to come out. Same thing here. So here we have the cephalon and the thorax are intact. We got a break in toward the end of the thorax, just before the pygidium, and so it may be that this one split its shell here and then sort of pulled back and then crawled out, or sort of backed out of its shell, tail end first, and then pulled its head out at the end. And so on. You can try and imagine these things. This one, the cephalon is pushed to the side, so maybe it came up and it went out this way. Here you've got one that the cephalon's kind of in place, but it's just destroyed this part of the thorax, and so it may be that it came out and it went in this direction. Yeah. But that's good evidence. And so trilobites, ecdysozoans, like all arthropods are, I think that's pretty easy. What do you do, though, when you've got a weird little worm like this? Is this an annelid? Because if it's an annelid, it's not an ecdysozoan, and so it won't shed its outer covering when it grows. It'll just grow the outer covering. If it is an ecdysozoan, it will shed. And so can we find evidence of this? And yeah, that's exactly what this paper is doing. So the first figure here is just showing you what this little worm looks like. This is Cricocosmia. That's right. That's what the C stands for. This is one we have several examples of this particular thing. It's probably fairly closely related to preapulid worms. It looks fairly similar, but it's in this ancestral group because it's not exactly the same. And weirdly, it's got these little plates that run up along the side, like little tiny bits of probably really ineffective armor. No idea if that is just there because genetically the thing that they descended from had those there, or if this is actually some kind of survival adaptation. Hard to see how, but yeah, whatever. I don't know. I don't understand. And so what they found on just a couple of these Cricocosmia worms is that you will get cases where it looks as though they may have been caught in the process of shedding their exoskeleton. Because, for example, instead of one set of plates, you'll have two, or you'll have actually what appear to be molts, just sort of outer coatings that are missing all the inner parts. Because these are very common at Chengjiang, and at Chengjiang you get preservation of everything, including like the gut down here. So here's a zoomed in version of this. So here there's only a single plate on each one, but here you can see two little plates that are offset by just a little bit from each other, which means it's grown another plate underneath and it's getting ready to molt. And that top molt has just started to come off and slide back. And sure enough, all the way down, both sets of plates on the right side and left side, you can see these little molts starting to move off by about the same amount. This is the molting process. You can see the digestive tract, so you can tell the actual organism is still in there. But yeah, good evidence of molting. Fantastic. Here's some more, and you can see this one has got, here we zoomed in. Look at that little double plate right there. And so you can see the old version and then these little FU, I think that might be a furrow, where the plate was as the new version is coming out. And they zoom in on it a little bit, but this is the new left plates. These are the new right plates. The old left plates have moved mostly off. The old right plates are still sitting on top. You can see the ones underneath are a little bigger than the ones on top because ecdysozoans shed their outer covering when they're growing. Yeah. So, yeah, of course it is. Of course it would be. That's how it works. Yeah, so there's a little bit about some evidence for the ecdysozoa. Most of these ecdysozoan groups we're going to cover in their own sections. They get their time in the sun. But we just thought we'd bring in some of these because we don't talk enough about Cricocosmia, and we got a lot of fossils of it, so we might as well, right? Thanks.